Okay, in this video, it's the wrong screen, we are going to learn about how to interpret HH-Pred data in the Pecan output and capture those notes and interpretations in our electronic notebook and Pecan notes. So there is a separate video that's associated with this assignment that helps you learn how HH-Pred works. This is going to be simply how to quickly interpret your HH-Pred results and record it in your notebook. So this is what your HH-Pred output looks in Pecan, and I just wanted to do a quick annotation of the information that you want to pay attention to in the table. In Pecan, we don't get the actual alignments of our query protein to whatever protein it's hitting. And so if you want to see an alignment and be able to identify important motifs that would, help support a particular function call, you'll need to run HH-Pred at the actual website. But a lot of times for easy function calls, and I would say that the portal protein is an easy function call, the data that is in the HH-Pred table in Pecan is plenty data to support the call. So the things you want to pay attention to underneath the column called hit gives you an ID for the crystal structure that we're looking at. The IDs that have these numbers that are a mixture of numbers and letters and the underscore and another letter, those are protein databank hits, and these are my favorite types of hits. Further down here, you can see hits to something with a PF ID number. This is to a different database that is going to have different formatted data. It's called PFAM. These are also really helpful, but we have to interpret the data in different ways. So pay attention to these IDs. Oftentimes the ID gives us a sense of whether we're looking at protein databank entries or not. The description of the protein is here. We really want to be looking for proteins that are from either bacteria or phage. If you see hits to human proteins, and you may get hits to human proteins, those are not likely to be super relevant to any function that we would be calling in a bacterium or a phage. And so try to stick to hits to phages and bacteria. This is where we can see that the probability score, which again, we are only accepting scores of 90 or above. This tells us the percent coverage of our protein. So 83% of our protein is aligning to this portal protein from SPP1 bacteriophage. This is the bacillus phage that's really well studied. It tells us which amino acids of the hit protein, so this phage portal protein, aligns to our protein. So it's aligning to the 31st amino acid to the 466th amino acid. This data doesn't tell us how big the protein is. So we don't know whether this protein is 1,000 amino acids long and it's only aligning to this part. So that is one flaw to just looking at this table. And then we see that our protein, which is the query, aligns from the 31st amino acid to the 462. There's also an E value here telling us that, that it's a high quality match. Sometimes we don't get enough information just looking at the Pecan table. And so it's important to run the protein sequence at the actual HH-Pred website shown here. On the input tab, you can enter a FASTA file. You can just put a protein sequence in, but I like to enter a little FASTA name here. You can see that I have a long list of hits recorded in the database, and I might want to go back and look at those. It's nice to see, oh, this was gene number 9 later on, because otherwise you get mixed up about when you're looking at your results. what gene protein sequence you entered in order to get those results. So try to keep track of what gene number, or better yet, a stop codon for the gene that you are analyzing. You're going to want to put in these 4 databases, and so you can just enter those in this window. There's a little drop down. It will automatically, by default, search the protein data bank, and I like to add PFAM and the UniProt database. I've also added the conserved domain database, but these top 3 are my favorites for searching. Then you would just push submit. This is just one, this is what I considered the most important alignment. This is the alignment to that Bacillus phage SPP1. It's a portal protein. I'm going to note not only the title of the hit and the protein data bank ID. I'm going to record that in my results. I'm also going to note that there's a 100% probability, and I'm also noting which amino acids are aligning to this protein. And so this is like a slightly different hit than the one that we saw in pecan, and I'm noting that our alignment is, the top is our query protein. The 17th amino acid through the 503rd amino acid of our query protein is aligning to the first amino acid all the way to the 501st amino acid of this SPP1 portal protein. And what's nice about this is it tells us how long the proteins actually are. And so 1 to 501 out of a 503 amino acid protein is nearly 100% coverage of that protein. So when we have such high coverage, we can be pretty certain that our query protein has all the functionality of this SPP1 protein. So that's really a high quality alignment. So let's just go to pecan, and I want to show you what that's back in pecan. Again, just like with the phage data, you can check off pieces of data that you use to support your call. Don't check things that you didn't use. Like, don't go check crazy and check all kinds of things down here just because they say portal protein. You're going to want to only check the pieces that you actually went in and investigated. The other thing, we'll talk more about this in a couple of weeks, but you can click on these links, whether it's here or at the HHPred website. So let's go to those results I was just showing you. I'll come back and look at that in a second. You can click on this link, and it's going to take you to a page and tell you everything about the crystal structure of that particular protein. You can usually see a nice little diagram. There is a reference. You can click here and go and read about the paper that published this particular crystal structure. I often will read through the abstract. And usually down here, you have to make sure you find the right chain that matches what your hit was to. So this is chain J, but you can scroll down here and let's see, is this chain J? Figure out which chain you are actually aligning to. I should have checked this out before. Here we go. Here's chain J. So this is the protein they are aligning to, but you can get a little graphical map of what this protein looks like and any annotations with it. Likewise, this is the protein. This is what it looks like at the HHPred. You get this visual that shows this is the whole protein that you submitted. This very first hit, oops, I didn't mean to click on it. This very first hit is aligning to the 33rd to the 465th amino acid of our query protein. You can hover over these and see what they are. So this first hit is to the portal protein of Mycobacteriophage BXB1. You can come down here and you see a list of these. And if you go a little bit further, you can actually see the alignments. If you want to quickly get to particular alignments, So let's see if I can find the SBP1 hit right here. If you click on the number, it takes you directly to the alignment, which you can investigate. So in my notebook, what I have done under HHPred, I've recorded that there's 100% probability and full coverage. And then to this particular protein, I just copied and pasted the protein databank ID number and its name straight from the HHPred website or from the hit in the Pecan table and popped that in there. Okay, that's it for looking at HHPred data for this particular protein. Again, HHPred data, this is direct evidence that the sequence and structure of this protein is consistent with the structure of known portal proteins. HHPred is the most important data that we can use to support a gene function, and it's dependent upon your interpretations, and unlike BLAST data, where we're depending on other people's interpretations.